much for coming. This is part 3 of our series on translational research. And today's panel is led by Dr. Christy Ahn from the School of Medicine. And I will let her go ahead and do the introductions for the rest of the panelists since we only have an hour. So thank you all very much for coming. And thanks to our panelists and to Christy.
Good morning, everyone. Thank you so much for coming. We're excited that this is the part 3 of our series. They've had 3 parts. I'm from the School of Medicine, and I also work for the Chancellor's Office, and I've been partnering with the DTMI, Virginia Burns, and Molly Starback in the Office of Postdoctoral Affairs to bring a series to you to help educate everybody about the translational research opportunities at Duke. And one area that we found is really important is how do you collaborate with clinicians? So before we have our panel discussion, I had a brief handout I wanted to go through to get you started thinking about some ways in which to form these collaborations, but also to kickstart our conversation and to take notes. Really, number one is networking to form relationships. And the panelists have a lot of anecdotes that they can share with you about how they really form those relationships, but it's really not just networking with business cards and getting names. It's really important to think about that. Going to grand rounds, attending clinically relevant meetings. Also, we want to highlight for you to contact folks in the DTMI. They are an incredible resource. And again, I'll mention Virginia. Virginia Burns is the point of contact. So if you have questions, whether it be for core resources or potential clinical collaborations, you just want to try something out and talk to her over the phone and get your voice warm, call someone in the DTMI. And also, if you met somebody who might come to part 2 of the series, any of those panelists contact, and also Barry Myers, who's a professor here at Stanford, you can ask them questions. They are connectors. Start asking them questions. Also, realize that being skilled, some of you may already know this, being skilled in these relationships and partnerships will go well for you, whatever career you will enter, especially if you want to do something different in regulatory affairs or medical science liaison work, anything like that, but this is an important skill and it's transferable. Also, we want you to think big. That's the thing. We want you to be able to explore and think of the impossible. Then when it comes down to it, just like the particular needs. And that really goes into number 5, and that's creating your elevator pitch, your BlackBerry window pitch, or your page pitch. Just something that really in that time in an elevator, or even on a BlackBerry where someone's looking, just fits in the window what exactly you're looking for. It's something that can take some practice and can be really useful. And of course, never ever give up. Be aware of people's schedules, but be prepared to reframe your questions and approach other people or review your approach, because something really will happen after this one, but it does take some work. And also, this kind of goes into project management, which can be an essential skill for you, whatever career you stay in, academia or outside, is that consider your SWOT. Really take it out and think about your career, but also your research partnerships and ideas. What are your strengths, weaknesses, opportunities, and threats? It's something that I've pulled out for both research and my work in administration. And developing excellence in project management skills is huge. It's something you can bring to these partnerships as you form them with clinicians or other teams, and it's a huge transferable skill, and it's a real asset to the team. And attending translational research training seminars, you're already doing that. We hope to offer another group of educational seminars next semester. We really want to know what you need and what your questions are, so please feel free to contact us and let us know what else you really need to know about translational research. And without further ado, listening to key messages today from the panelists, a panel of experts here to share with you, answer your questions. We really want you to feel very open and asking anything like, what's a text page, and should I be using it, and how long should it take before someone answers an email to me? So with that being said, I invite the panelists to start with Dawn to introduce themselves, give their background, and then we'll get into our discussion. So Dawn, welcome, and thank you for coming. So let us know about your background.
Thank you, Christy. My name is Dawn Bowles. I'm an assistant professor in the Department of Surgery, and I've been here for pretty close to 6 years now. I was recruited in the Department of Surgery from UNC Chapel Hill Gene Therapy Center. And the predominant reason why I was recruited was the skill set that I had acquired there for viral vector development. and there was a large group of individuals, there still is a large group of individuals in the Department of Surgery who have an interest in gene delivery for different forms of cardiovascular disease. And that's predominantly my area of focus.
My name is Herman Statz. I have been at Duke since 1993. I first came here as a postdoctoral fellow in the Department of Medicine and stayed there for a number of years as a research track faculty member, and then I switched to the Department of Pathology, where I'm now a professor. And my lab has always focused on mucosally administered vaccines. When I first came here, I worked as a postdoc in Bart Haynes' group working on mucosally administered vaccines for HIV, and I continued to work with Bart over the years. And then my research group now, although I'm not funded for HIV vaccine research, I'm still continuing to evaluate intranasal immunization, and we have been funded to do that for anthrax, botulism. We have a translational project now funded with Dr. Sunday where we're evaluating intranasal immunization for influenza. So my lab, really, I don't really care about the pathogen so much. We also are interested in intranasal vaccines for cancer. So my research is predominantly focused on utilizing needle-free methods of immunization and optimizing adjuvants and formulations for their delivery.
So my name is John Sunday. I've been at Duke since 1991. Came here as an intern, and I came here actually with training from the medical school as a bench immunologist. And when I completed my medical training, I actually was also a postdoc in Bart Haynes' lab, and did that for a few years, and then really evolved into more of a clinical trialist and a clinical researcher over time. My background is in rheumatology and allergy immunology, so I really try to apply that immunology background to clinical trial work. And so really over the last few years, I've worked across the therapeutic areas with a number of compounds, from preclinical to phase 1 to approval. I had a great opportunity to actually work on that progression, working with my first field on the drug Krystexxa, which was actually approved for treatment of refractory gout, approved September 14th. But I remember sitting in the fellow workroom while he was writing his small business collaboration grant to actually get this project going 12 years ago at this point. So that's the kind of timeline that many of these projects are on, but has resulted in an approved project. Mike is, I think, one of the key people here who's done this twice. There's actually developed 2 drugs that are approved for use humans now as well too. So it's been a great situation for me. It's been great to work with Herman, and I really would like to echo the networking comment too. Just the people that I've had a chance to interact with over the years in different capacities has really yielded fruit for me. Sometimes 5, 6 years down the road here as well.
So we want to ask, and feel free to anybody jump in, what's the best way to start thinking clinically? So beyond maybe you might be working with cell lines or tissue samples, but really getting your mind around hypothesizing a clinical problem and starting that collaboration. What's the best way to start?
In my experience, mainly the clinicians approach me with the question. They have something that they've seen in the clinic. For example, Dr. Carla Milano, my primary collaborator, performs the majority of the transplants here at Duke, and one of his main areas, or heart transplants I should say, one of his major goals is to increase the number of hearts available for transplantation. And in fact, we have a project that has just been recently funded by the DKRI to do exactly that. So he comes, to our group with these concerns, with these problems, as opposed to me personally as more of a basic scientist coming up with these questions.
So from my point of view, I think I have always been interested in working on a project that would lead to a product for humans. But I think from my point of view, in my experience over the years, is I've had to realize that a lot of the work that I started originally in a mouse model once you have done this for a number of years, you realize that the results you get in a mouse or a rabbit or some other animal may not really translate to application in humans. So although have always been optimistic and enthusiastic about the work, it took me a number of years to really realize that I have to step back and ask myself, is my research really ready to go into humans? So, because I think in my own case, if we had taken results from our first mouse study and went into a human system, we probably would have failed miserably because we did not really understand all the fine details, and there's a lot of difference between a mouse nasal cavity and a human nasal cavity. So over the years, through continuing our research, like John and others have said, it takes time. It really does take time for some of these things to develop. And over the years in our animal models, we've understood some of the technical details and requirements for making intranasal immunization work better in animals other than mice. And that's finally led us to the point where we are now doing more studies in rabbits, monkeys, and finally in people, that although all of us may have a great interest in doing the human studies or the clinical studies, your research has to be at a point where it's really ready to go there, because I think if you go there too soon, you run the risk of failing and then possibly getting a reputation that your research really will not work, when in fact it may work if you understand the details of the situation better. So I think one thing is time and be patient, but stand back from your research a little bit and ask, is it really ready to go into humans, and do I know enough about this system to be successful once we get into human studies?
Yeah, I think that there's probably a couple of ways to approach this. I mean, I think the interaction can happen from either direction, that you have an idea that you think is ready to go into people, or that you would like to actually use human samples or specimens to study. Or somebody may approach you, and they've learned about the work that you're doing, and they think that there might be an interesting clinical application of it, and so they might approach you as well. So I think that one of the key things you can do is put yourself in position to either be the recipient of or the initiator of those kinds of interactions. And so, and it's one of the things that we're actually trying to facilitate here as well in the Translational Medicine Institute. So my key role now is actually serving as the director of the Duke Clinical Research Unit, our early phase first-in-man unit. And one of the things that we are trying to do is to work with the DTRI to figure out ways that we can facilitate this interaction between laboratory investigators and people who have an interest in this kind of early phase or proof of concept work. But attending grand rounds, attending clinical research seminars, the Duke Clinical Research Institute's weekly seminar, I think is a great way to understand what's going on in the clinical world. If your particular field of interest is relevant to, say, nephrology, then attending nephrology grand rounds, establishing relationships with the clinicians there is a great way to understand what may be relevant, and number two, to have those relationships in place so that you're kind of a known face as well. Certainly, it's incumbent upon the clinicians who are interested in this kind of work to also do the same thing too, and attend, the immunology research conference or some of the things as well. And so that's certainly an important aspect of that. But I would say be an initiator of that contact is a great way to do it. It will give you ideas on how to maybe translate your work, and give you ideas on who you may partner with to do it, and to make it easier to strike up that conversation as well. So I think that that's an important piece. And then hopefully, I think you'll find that the DCRU and the DTRI are ways that we can maybe serve as a clearinghouse for those kinds of interactions as well, and to facilitate those interactions. So that's a key aspect of this.
So I wanted to follow up on what you brought up about connecting the researchers to this first phase research. How should they contact you? Should they contact you, or what's the process? Is it an email or phone call? I'd like to know a little bit more about that.
So would you like me to answer that first?
Yeah,
okay,
sure.
Right. So, I think it depends. It's going to depend on the person, just like anything else. But I mean, certainly from my standpoint, I'd love to hear about new ideas and ways that we might try something. So I personally am always receptive to this. I think that if you have taken some time to get to know who the people are who are in your field of interest, you can understand a couple of things. And here's some things that I think are important to know about the clinicians is what they do. Some people are in the clinic all the time, and they actually may not have an investigative career. And so So it's going to be difficult for them to devote substantial amounts of their time to actually interact with you or work with you. So you probably need a clinical champion within that division or clinical therapeutic area to maybe help bridge that world. The more somebody spends their time in a clinical setting, the more they begin to view their world in 15-minute increments of time. And so you have to just understand that, that mindset, which is really different. So, say, hey, well, let's, get together at 2 o'clock and chat about this idea is probably not a feasible thing. It might be feasible to get together at 7 o'clock, or it might be feasible to get together at 6 o'clock, p.m. or something like that to talk. So I think that, just understanding the world they live in. But a lot of people who are, consider themselves clinicians, like I do, still spend about 75% of my time doing research. So that's a little bit different game. So understanding the world that those people live in is going to be helpful to you and understanding how to approach them. But I think an email is is probably an easy way to make contact. If you have been participating in clinical conferences, then it's an easy time right after conferences to strike up a conversation as well too, or to give that elevator pitch and say, hey, can I follow up with an email, or can we arrange a time to get together? And I think for most people who are interested in clinical investigation, they're going to be very receptive to those ideas as well. So I think that's a great way to kind of break the ice.
And please feel free to interrupt us at any time to ask follow-up questions and dig deeper. It's welcome. Any additional comments about that first approach?
In terms of not necessarily making the initial contact, but actually continuing the collaboration, I think it's very important to understand, as John mentioned, the concept of time that the clinicians work under. You need to be very flexible in your time, basically your time management. You can, if, for example, Dr. Milano in between cases may call and want to talk about a project. So you may go to the cafeteria and have a lab meeting, have a mini lab meeting. You may meet after hours, before hours, that kind of thing. I've gone to different hospitals to meet with residents about different projects on Saturdays and Sundays and things like that. So I think for the most part, surgeons do operate, or their time scheme is basically 24/7, so you also need to be very accommodating to that kind of thing if you want to continue to work forward with these individuals.
Any additional?
Well, I think one thing that you have to keep in mind again is this third point you had on this first list here of realizing that being skilled in partnerships and relationships is important for wherever you go in your career, and also developing these collaborations with clinicians or either other basic scientists. You have to realize that if you have a reputation as being not productive, not collaborative, not interacting well, that people will not want to work with you. So I think part of networking, in my opinion, that has been helpful for me is that I think over the years I've worked with enough people here at Duke that, for example, with Bart, people have gone to Bart and say, Bart, can you help me do something? And if he's not interested or doesn't have time, and if he thinks it's something that I could do, he'll say, well, contact Herman. And that's happened several times. It has led to large grants being funded. So you have to be a good citizen. You have to have a good scientific reputation. You have to be productive. And when you have that reputation, the networking gets easier, because if people ask about you, you'll have a good reputation, and that is clearly helpful in my mind in getting that network started.
Not only though in networking, I think also in being a member of a team. It's very important, because you really want to work with people that you like and who like you, and that really makes things go forward faster. It doesn't put any impedance and doesn't impede the process of the project.
Yeah, very, very important.
Are a lot of these things and tips that you're mentioning, I see it in the faculty level. Anything different, or how should a postdoc be viewing it? Or is it the same? Did you do these things as postdocs?
I personally, no. As a postdoc, I did not do any of these kinds of things. As a postdoc, I was very much focused on one project and one project only. Narrowed tunnel vision, went to the lab, did my work, that kind of scenario. when I obtained this position, it was absolutely necessary for me to be able to network with a lot of different people. I mean, I basically came into a department where there were 9 or more PIs who wanted to work with me. So I was, I needed to be able to interact with these different individuals and get things done. And it's been a learning experience, but it's also been a very fun experience, interacting with many different people. And the one thing I can just say is don't be fearful about contacting people. Don't be afraid of talking to them or, setting up the initial conversations, because most often, most people are very receptive to listening to you and to your ideas. And here at Duke, I can say very helpful.
Yeah, I would agree too. I mean, I think that obviously how you spend your time like that is something that is important to work out with your mentor if you're, a postdoc. But I think that certainly in terms of interacting with the clinicians, we are with medical students and residents all the time, and so I think that that's part of the reason why we're here. So I think that you'll find for the most part that clinicians are very comfortable with recognizing that not everybody's a full professor, and so so it's fine to interact well. And in fact, I think it's enjoyable. And I'll just give you a great example. We had a endocrinology fellow working with Mark Feinglass follow up on a very casual hallway conversation I had with Mark Feinglass. He showed up with a great elevator pitch, one day to me, and we were able to turn that into a little pilot project funding grant from the DCRU to help get a clinical project off the ground. And the key thing was, I think, gets back at Herman's point. So not only was this person willing to go out there and network and kind of put it on the line, but they also followed up promptly with a great protocol concept sheet. They'd done their write-up. They were clearly demonstrated that they were committed to following through with the project, and so that made that a really easy decision. So I think that a good networker, but no meat on the bone in terms of productivity, won't get you very far. But when you can merge those two together, that's a real good recipe for success, I think. So.
Christy, I have a question. I think so. You just raised a point that was sort of cogitating. You all are talking about things that are not traditional sort of consciousness in an academic research environment. You are talking about networking and elevator pitches and informal collaborations where we step up and introduce ourselves. And I think that that's enormously important to raise for a group like this, to build confidence and to recognize that this is not a time to relax and rely on traditional means of advancing our careers. In a more traditional, sort of to reach back into the traditional milieu, what is the role of journal clubs and the literature in informing young investigators and helping them know what resources are available to them in this environment and beyond this environment, and what kind of, how important is it for people who want to advance an idea into a novel application to really do their homework and know what investigators that have come before them have done and be prepared to sort of review the literature and seek out, rather than just saying, I'm Vicki, I'd like to talk with you. I'd like 15 minutes of your incredibly valuable time. How eloquent would a first introduction need to be, and how comprehensive an awareness of the landscape before you have a conversation like that?
Lead takers.
And what would you do to become sufficiently eloquent?
So again, that gets back to my comment that some of these things take time and you have to be productive in the lab. So I think for me, I would prefer that the person come to me with some understanding of the background of the model that we want to do. But sometimes your first interaction may raise issues that you were not aware of. So quite often there may have been times when I've gone to someone and say, I want to do this, and they'll sit down and they'll say, well, that's a good idea, but have you considered A, B, and C? So sometimes that first interaction is a learning experience for me that I have to go back and think more about, am I really ready to do what I think I want to do? So And that's just part of a good science, I think, is being prepared and having your background ready. And sometimes things are ready to go forward, and sometimes they aren't.
I think that's a really good point. I think that you should feel free to, I mean, if you're trying to think about this in terms of interacting with the clinical people, to use them just the way you would a colleague down the hall if you're trying to figure out how to engineer a construct into something that you're trying to transfect or something. So use them as a methodologic or an idea generator as well too. So I think that being familiar with the field is important, but also saying, hey, I want to just kind of pick your brain. Here's something I'm thinking about. Do you think that there's an opportunity here? I think that's a perfectly good conversation to have. So I think that it's always nice to be prepared, but at the same time, I don't think you should feel like you have to have a finished product that's ready to write the IRB protocol and start the next day. I mean, I think it's fine to really let that evolve over time, because usually it does need that kind of incubation for quite some time. So.
Thank you for your question. It's definitely understanding the difference between a perfectly packaged pitch versus warming up your voice, having a chance to vet it, maybe emailing the panelists that you all know, or Virginia, giving her a call. And I will go right into this question that actually came from the audience. This is a good way to start this. Someone asked, I'm interested in getting human tissue samples for RNA extraction. Is there a process for acquiring samples or setting up collaborations with surgeons? This is an excellent example of using a resource now so that we can put our minds together and answer you and direct you in the right direction so that you can then hit the ground running. So let's use that question to piggyback off of Vicky's comment and start with the panelists.
Call me.
There you go. That was easy.
We have a human heart repository, which we would be happy to collaborate with individuals. So just give me a call and we can set that kind of thing up.
What about different tissues?
Just hearts. Sorry.
Okay. So if it's something
you.
different, how would somebody go about this collaboration?
I think you have to kind of think about, probably consider a couple of factors in that. Number one is, can you find somebody that has an interest that overlaps with yours, so that there's a opportunity for academic collaboration in that project, and then get an understanding then of what actually would be involved in obtaining those samples as well. So certainly the first thing you want to do is identify resources around you, like a tissue bank or something where you can, there's stuff already in the bag for you as well. So that's obviously step one. But if you're in a situation where you say, no, I really need to, I'm starting fresh here, and every repository had to start fresh at some point. So the question is how do you pull this off? Understanding what's involved in terms of the IRB process and the timing involved and the sample processing and preparation. Thinking about how accessible you'll be when a sample is either available or when the kind of patient that you want to get a sample from happens to show up to the clinic. So there's a lot of variabilities in this that you'll want to try to work out. But where I see it not work out well is where you're looking for a serum sample from a particular patient population. There's not necessarily an opportunity for the people who have those patients to actually collaborate with you in a way that's going to lead to grants or publications, or maybe they're not even doing clinical research in that area, and then hoping that you're going to get called to come pick up that blood sample or anything. That probably is not going to work. So it's really trying to understand the process, forming collaborations, creating situations where it's a win-win for everybody who's involved, because the price tag of actually getting an IRB protocol through, obtaining the specimens is pretty immense, and the cost of clinical research is just staggering. And so figuring out ways to overcome that is a really important part of being successful in obtaining those samples.
I think that's an excellent example too of doing your homework but still leaving some things open. So you know all of that, you know what it takes. So go ahead.
I have a couple of questions. One, you're talking about collaboration and two very different approaches. And so do you have any suggestions for when you kind of are stuck at, they have clinicians have one goal or objective and you have another kind of a goal? How do you work that out? And are there people, like would a clinical research coordinator even understand both and be able to bridge that? Or, who would you use to help kind of in the middle come to an agreement. And the second question on that, on funding, are there specialized grants, you know, for clinical research, or would you kind of go through an R1 or a clinical grant, or go two different questions?
Well, I think probably, I think that probably one of the things getting at the first question where maybe the goals are different, I think you have to look at those goals and say, you know, which of those goals are different. Is it a timeline goal, or is it a scientific direction goal? You know, because some of those things can maybe be modified or merged over time. Some things that a clinician might want to do based on the work that you do just may not be feasible, and so it's part of the kind of a cross-education process, and vice versa. Something that you might be interested in obtaining is maybe just not, you know, feasible to do. Most of these things, to be successful, are going to require somebody besides, you know, you and the lab and the and sort of a co-PI that you might be working with, like in terms of a coordinator as well too. So understanding the resources that that investigator has available to them to work is going to be important as well. And that's again another example of where I think that maybe with the resources in the DTRI and the DCRU, where some of these things are already, that infrastructure has been established, if we can figure out ways to pull in and connect the you know, the laboratory person with the clinical person, is where we're going to be successful in maybe overcoming some of those problems as well. We're in the process of actually trying to create you know, what we want to call clinical research incubators, where, you know, a person can come in with a research idea. We can pull in people with expertise in that field, and you know, have a little, you know, 1 hour seminar, you know, or conference roundtable to talk about how you might approach those problems. And hopefully that's something we'll be able to roll out in the DCRU over the next 4 to 6 months here as well too. So.
For the additional question about funding, does it take a specialized grant or R1s?
Roman's lived this world a lot, I know. So.
Well, you know, although there is an emphasis from the NIH on translational research and things like that, I think you just have to be persistent from the funding issue and write investigator-initiated grants, whether it's an R21 or an R01, or look for program announcements or look for local funding. I mean, unfortunately, a large part of our jobs these days is identifying funding sources, and sometimes they're R01, sometimes they're R21, sometimes they're institute pilot grants. You just have to keep your eyes open and search for them.
I think the DTRI, through their pilot program, offers very valuable start that enables you to jumpstart to get additional funding, and definitely I would take advantage of that, either through the pilot or through even their core voucher program. Will enable you to connect with a clinician, come up with a small project, and I believe postdocs can also apply for this. Is that correct? The vouchers, and that gives you some seed data that enables you to move that forward and write a pilot project, and then maybe move that into an R21 or an R01. So definitely the DTRI is an excellent resource.
So I've been to a couple of the DTRI seminars, and I checked out the webpage. And so I'm a biomedical engineer, and most of the mechanisms and things I've seen have been more about drug or maybe genetics. Is there any type of mechanism or foundation here for someone coming from an engineering background? And if so, what are
Coulter. Do you
Coulter,
guys know about
yeah.
Coulter? You explain
So
that?
there's a funding program. I don't really know its name. I just know it as the Coulter.
Coulter Translational Partnership Program. It's a soon to be a $10 million endowment by the Coulter Foundation.
So although my interest, as I mentioned, is vaccines, a number of my colleagues got together last year and put in a Coulter proposal where we would do some nanoparticle-based vaccine formulations that eventually did not get funded, but that was a combination of the immunology vaccine research with biomedical engineering for nanoparticle formulation, and we came together and put in a proposal for that. So the Coulter system on campus, I think, would be suited for what you're talking about.
The person you should write to or call is Melda Ouzbil. Have you met Melda?
I have not.
Z-B-I-L, Melda.
Okay. Melda Oosville. Okay, great.
And sort of to follow up on Herman's point, so Herman and I have one of the pilot project programs through the DTRI right now, and I think we applied probably 3 or 4 years consecutively before we got funded, and I was lucky enough to be invited to participate in this Coulter application last year, and it's our intention to wear them down too, and ultimately get that funded. So that's, I think that those are the kinds of things. It's persistence, I think, is also helps.
One of
Can I
the
add something beyond persistence is that the folks at Coulter have an amazing advisory board. The people at the DTRI have not only resident experts across lots of disciplines, but access to expertise across the whole ecosystem and outside of the ecosystem as part of their advisory board. So you're not just wearing people down, you're actually honing your proposal. And I think that it is a way to learn about the ways to avoid the obvious pitfalls. Those pitfalls that slow you down in a voucher application or a pilot application are also going to slow you down in an R21 or an R1 or a, any kind of grant application. So it's a way, it's painful, but it's cultivating good judgment on your part, I think.
Absolutely.
I might want to add just one other thing. So one of our pilot projects is actually involved in making an ex vivo heart perfusion apparatus. And we were basically told that we were lacking an engineer on our team. So, I think if letting the DTRI know of your interest in these areas, they may be able to kind of match you up with different teams that may have a need of someone with your expertise.
Does everybody know, Vicki, I'll put you on the spot, as she introduced herself, does everyone know her? Vicki, were you key resource?
I'm Vicki Christian. I work for Rob Caleb and Bruce Solinger and John Sunday, and this is Wesley Georgiana. This is Virginia Burns, who haven't been introduced today, but Virginia ran that seminar last time. They are project leaders who are doctorally prepared scientists who have chosen careers that are principally translators. They're principally people who are sort of intentional team scientists who are not pursuing original research goals, but are working to build teams and advance the ideas and cultivate and hone the ideas of other people. So that's what we do. And the website is evolving. I saw Virginia cringe when somebody mentioned going to the website. You did. But I think it's safe to start with Virginia or start with the website. There are names and email addresses. If you type in cores and you want to know whether there's a viral vector core, you're going to ultimately get sent to Virginia, who will send you to Dawn, and we're going to try and make that more interactive. Just so people know, I started at Duke, worked here for many years, went out to the commercial sector where I learned other things, brought those things that I learned over 14 years in the commercial sector back to Duke when the CTSA grant was funded, which the director of which is Rob Taylor, who gave the first seminar. Some of you might have been here. But that's when I came back to Duke, and by having the freedom to recruit people like Wesley and Virginia, I'm able to, it's a dream. So, thanks.
Thank you. And I know it may not be your favorite thing, but on behalf of the panelists and myself, before we wrap up, can you all introduce yourselves, like what department and maybe a research project, so that we can all hear and maybe start that conversation? Can I start with you?
My name is Sandy. I'm from the Department of Radiology.
Okay.
Are you a postdoc?
Yes.
Go ahead.
Oh, I'm Acharya from Radiation Oncology.
Postdoc?
Postdoc.
I'm Josh Feinstein. I'm a grad student in the Department of Pathology. Herman's actually on my committee.
All right. He's here. Check.
I'm Mary Sobedakur, and I'm working in the Division of Infectious Diseases. And I'm not a doctor.
Okay. Follow?
I'm Bala Balakumar, and I'm at the IGSP and research assistant.
I'm Ginger Reed. I'm a graduate leader in the Department of Immunology.
I'm Emily O'Corin, and I am also a graduate student in the Department of Immunology.
I'm Joel Mainland. I'm a postdoc in molecular genetics.
I'm Julie Wilmington. I'm a grad student in engineering. Are you considering
Peter Alexander, a postdoc in pharmacology.
I'm Katie Zaraka. I'm a grad student in anthropology. So I see in postdoc in human vaccine study and hopefully get the rest of my trials.
Andre Snelling, postdoc in biomedical engineering / kind of neuroprosthetics.
Hi, I'm Patrick Rose. I'm also within UTMI. I started about 2 months ago, so I'm looking forward to things with you. But I've had a lot of industry experience as well in clinical research and drug development, devices and biologics. I'm working right now with Dr. Marilyn Miranda at the Children's Health Environmental Health Initiative. I'm really happy to be a resource for any of you.
I knew I recognized you. Out of context.
So with that, so you know your audience now, big tips for graduate students and postdocs interested in translational research, both what you can do as a graduate student and postdoc. We could focus on the faculty trajectory for this part. Any tips?
For me, it was being at the right place at the right time. That was it, with the right skill set.
I agree with that. I think from my experience, it's hard for a graduate student to really get deeply involved in clinical studies because sometimes they're unpredictable. Sometimes they take a long time to get things set up. So I've had experiences in the past where grad students came in hoping to work on a clinical project, and it just never happened because there were so many delays that it was very risky to have in my opinion, to have a graduate student solely focus on clinical or translational studies that are dependent upon the clinic. So I think that's difficult. It might be easier as a postdoc to go into a lab who has those types of studies already established, where you can step in and get the experience and get involved in them in projects that are already established. But being at the right place in the right time, again, goes back to what I said earlier, being a good laboratory citizen, being a good citizen of your institution, being productive, because that helps build your reputation so that when people think of you or when your name gets mentioned, there's a positive association with that.
Should they be asking, I'll just interject this, to shadow or have somebody clinical on their committee if they can? Is that important, especially graduate students?
I think it depends on the project, in my opinion. I mean, it certainly can be extremely helpful.
I think learning the field, learning the clinical field, and taking time to do that might yield more fruit than, say, a shadow experience where it might be fairly piecemeal, and you might actually figure out a way to really target your shadow experience in the clinic once you begin to understand the issues that are important in the world of neurology or stroke biology or whatever application there might be to your work. So. And I think that's attendance at grand rounds, reading, through some of the clinical journals, understanding what's happening. Nature medicine is just a great way to review the field across at a high level, so you don't have to worry about, clinical trial design, but just sort of thinking about what may be an important advance. It's just an example of that. When you can attend the clinical meetings, I certainly know that in my fields of rheumatology and allergy, we clamor to get laboratory science presented and brought to our meetings. And in fact, especially for postdocs and graduate students, usually money available for travel expenses and things like that, just to try to encourage that interaction with the clinicians. And so, by doing that, you begin to understand what is important in a particular field or therapeutic area. And I think you find usually a welcome audience for your work at that area. And so I can give you an example. The American Academy of Allergy, Asthma, and Immunology. I know a couple of you are in immunology. Would love to see that kind of work brought. And I sit on the annual meeting planning committee for the Quad AI, and it's a key issue of ours is to advance the amount of that work. And it can even be secondary presentation of data as well too. So depending on the society, it doesn't have to be the primary place that you presented that. So if it was, American Association of Immunology was where you first presented that, but you also want to send it to the Quad AI, it will usually be acceptable. And so you just have to read carefully about what the abstract rules are for that as well. So it's a great way to network and to expand your connection into the clinical world.
Any current challenges for any audience members that you want our expertise, at least right now, trying to translate or work with somebody in the clinic that we can think about for you? Any questions?
I have a question about ownership. When you collaborate with someone, how does it work when it comes to write a publication? So you may be supervising graduate students who need to be first authors on papers, and the clinicians may be supervising residents or fellows who also want to be first authors on papers. So how does it work exactly? What can we expect from collaboration?
It's a good question.
You need to have a good conversation beforehand. So not when the paper is getting ready to be submitted, sit down and say who goes where, but that's, in my opinion, has to be part of the planning up front, understanding the role of each party involved and how it may evolve into a publication. You need to have some kind of discussion like that up front, especially if it's someone that you're working with new. I mean, the stuff that I've done with John, we don't really have a set discussion like that up front because I have known John for so long that I know that we'll sort it out and we'll be happy with it. I have no problems concerned with that. But if it was someone new, I'd want to have some kind of a discussion up front to make sure that we discuss that issue.
And a perfect project has opportunity to spin off, different kinds of publications that, one group can take the lead on and the other group can take the lead on perhaps the other as well too. But definitely pointing out where you have trainees that you're responsible for and how you're trying to work to advance their career is really important to build that into the discussion early.
Another thing I might add is to have, if you can, a pretty diverse research portfolio, because translational projects take an enormous amount of time from start to completion, and so you really need to have additional more short-term projects to show your productivity in terms of publications.
And how is the AP&T process looking at team science and publications? How is that?
I can't really answer for that question right now. I think that there needs to be, and I think Dr. Califf may have stated this at his initial talk, where the climate needs to change. I think that being a part of a clinical trial with your name in the middle of the publication should receive a nice, you should be able to get points and consideration for that. And I think that the times are changing, but probably not as quickly as they should be. I don't know, maybe you guys can have more comments about that.
Well, I think Dr. Haynes has said the same thing when he was first getting the Human Vaccine Institute developed. A lot of the comments that he made to the members of the Institute as younger faculty members was that it was a little bit different and that it was going to be a lot of teamwork and team building, and the academic centers needed to realize that type of science was going to be different than the historical, you write an R01, you publish a paper as the senior author, and you get recognized for that. So a lot of these huge team efforts, the institutions should recognize that is a different type of science, but it's still a measurement or indication of your productivity, and there has to be some recognition of that.
I think among kind of the top tier institutions, Duke is probably more forward looking on this than many. And so I think as you kind of move about the world and move on with your career, it's important to understand how whatever each institution looks at that, because it may be different. And I think that some of the infrastructure and resources that you see here at Duke are things that don't exist at other places. So it is evolving, but it is important that you kind of do your homework as you think about your next moves as well.
One other brief comment. I do know that the NIH itself and their tenure-based faculty have made changes in how they are recognizing team-based research. They're now having those add to the portfolio, experience in teams and publications based on team team-based science are being recognized by the NIH and their tenure track record.
I believe it's coming.
Yeah.
Any more questions from the audience at this time? Well, I want to sincerely thank our panelists for your expertise and your time. It wasn't 15 minutes, it was an hour. We were very lucky to have you, so thank you very much. And please don't hesitate to contact any of us to help point you in the right direction. Any question. Thank you.